Reagan Walter, and the title of my project is Increasing the Solubility of Fertilizers in Soil to Avoid Presence in Runoff. So, fertilizers are essential to the agriculture business. However, fertilizers contain a large amounts of chemicals such as phosphates and nitrates, and these are used to aid the plant in its growth. However, high quantities of these chemicals are not always absorbed by the plant and end up making its way into runoff, which then goes into oceans and other bodies of water. And this results in problems such as red tide and eutrophication. So my idea is to construct an experiment that tests which component can most efficiently reduce nitrate levels in runoff. And significantly reducing nitrate levels in runoff could lead to decreased frequencies of eutrophication, red tide, and other forms of water pollution. So to discuss, red tide is a harmful algal bloom that occur along coastlines when nutrient levels are imbalanced, and it's estimated that during a red tide, nearly 100 tons of fish die per day. This is not only very hard on all of the marine life, it's also difficult on humans as well because they can develop respiratory issues. And eutrophication is similar to this in that it also deals with the imbalance of chemicals coming into the water, and so either a complete body of water or portions of it can be supplemented with too much minerals, and it causes decreased lucidity through the water and creates anoxic conditions, which is very harmful to all of the marine life living in the oceans and bodies of water. And so this is EP Creek, which is a creek that runs through the majority of Yanoga, the Yelza State in southern Nigeria. And it's used by much of its population for fishing, bathing, cleaning, etc. And recent studies have found that there's been a lot of waste dumped into this creek, and further studies found that this is from chemicals from runoff that have led into the creek. And I wanted to include this just to help give sight of a problem that's happening currently that's affecting humans, because this creek is very instrumental to the population. And then a group of researchers created an engineered bioretention to improve nutrient efficiency and the environmental impact of fertilizers. And they used alfalfa, sawdust, composted leaf mulch, wood chips, sulfur, and straw to test the denitrification. And they actually found that newspaper was the most sufficient in reducing nitrate levels, which actually led me to use newspaper in my personal experiment. So this is grass buffer strips here, and after reading several articles, I found that grass buffer strips are vegetative areas surrounding a body of water that is used to capture pollutants, and it helps to remove pesticides, phosphates, and nitrates from the runoff water. And this just offers a simple solution in reducing the chemical runoff into lakes and rivers or creeks, and it just helps to capture any heavy chemicals that would be escaping from crop fields. So with that being said, there exists a gap in the research where the effect of nitrate-reducing components on nitrate levels in runoff is not known, and it is the goal of this research to fill the gap by studying various components on nitrate levels in runoff. So my hypothesis is if pine pellets, alfalfa meal, and newspaper are tested to remove chemicals in runoff, then pine pellets will be the most efficient in reducing nitrate levels. I think this because pine pellets were actually originally created to be used for animal bedding, and they were made to absorb animals' urine, and since I was looking at trying to find something that could help absorb water, I thought that this could act as the same and hopefully absorb the water and hold the nutrients that would have normally escaped. And so to do this, I designed an experiment, and I constructed bottles. I cut the bottles about 2 inches down and then flip them over inside of themselves, and the top half of the bottle is going to hold the plant fertilizer and component, and then the bottom half will capture the water that will mimic a runoff. And then 5 holes were drilled into each bottle cap, and all of the bottles were labeled. There was 3 components and 3 trials for each component, and then I had my control, of course, so there was a total of 12 bottles. And each bottle's received its corresponding component, which was newspaper, alfalfa meal, and pine pellets, and then all bottles received the same fertilizer and 2 arugula seeds as well. And the arugula seeds weren't necessarily important in this experiment. However, I wanted to have a plant to see if any of the components had an effect on the growth of the plant, just to see if there was anything that could harm it. And then for 22 days, each bottle was watered 5 times a week. And once this 22 days are up, 10 milliliter samples were taken from each bottle, and the nitrates were tested using a Lamont colorimeter 3 and nitrate tablets. And here's an example of what all but the pine pellets trial actually looked like once they were done sitting for 5 minutes before being scanned into the colorimeter. So here's an example of what my bottles look like. These are my 3 controls, and these are my 3 alfalfa meals. And then these are my 3 pine pellets component and my 3 newspaper. And there's not much water in the bottoms, but as you can, this is just because this was like the first week, I believe, in testing, and so I just wanted to give a visual so you can see that how it was designed, but you can see that the plants are growing and the fertilizer is in, and then the water is just slowly being collected. So to discuss, this is my data table, and as you can see, I have my 3 trials and components, and then my average, standard deviation, standard error, and a % from the control. And if you look closely, you can see that the pine pellets decrease nitrate significantly compared to newspaper, alfalfa meal, and the control, with over a 97% decrease. And to put into a more visual, with my bar graph, you can see the pine pellet has a significant decrease in reduction of nitrates. And alfalfa meal and newspaper did also have decrease. However, compared to the pine pellets, it was not as significant. And their standard error bars were included, of course, to offer reference to errors that might have been made. And so to discuss, as I just mentioned, average, standard deviation, and standard error bars were calculated over each component. And compared to the control, the newspaper showed to have 69% less in nitrate, pine pellets had 97.2% less in nitrate, and the alfalfa meal showed 41.4% less when reducing nitrates. And so the average final nitrate concentration was significantly different across all groups when run through the ANOVA analysis. And the pine pellets showed significantly less nitrate than the control when compared using a 2-sample t-test. So for observations, I wanted to include this because all of my alfalfa meal bottles had mold growing in the fertilizer, and the water that was being collected turned a yellow-brownish color, and I believe that this was from the over-excessive amount of moisture that the alfalfa meal caused, and from the dyeing of the color, which led to the color in the water. And then, as I sort of mentioned before, when I was testing my samples, all but my pine pellets component had turned bright red after the nitrate tablet was in the sample for 5 minutes, and this is likely due to its absence of nitrates compared to all of the other components. So to conclude, pine pellets have showed to be the most efficient at reducing the nitrate levels, which did support my hypothesis, and adding pine pellets to fertilizers could have a significant effect on decreased frequencies of red tide and other water pollution problems if implemented in the right way. And then I wanted to mention that potential errors could have been made when I was getting my results, because the colorimeter did test out of range for all but my pine pellets component, and so each trial had to be diluted with distilled water, and so I did my best to make that correct dilution. However, there could have been errors. So to readdress the gap, as established previously, a gap aimed to be filled by adding a nitrate-reducing component to fertilizer. This gap was resolved by adding pine pellets, which reduce nitrate levels in runoff water by more than 97%. However, limitations still arise. The environmental impact of pine pellets are still unknown and have not been tested in the agriculture field. Future implications of pine pellets in fertilizer could lead to further conclusions on its ability to be implemented to fertilizers. So for future implementations, newspaper and alfalfa meal would likely not be a suitable candidate to implement into fertilizer. However, I believe that pine pellets do have potential to be a suitable candidate, and I think that they could be implemented in two different ways. They could either be added similar to a grass buffer strip, where to a location where the water runs, and they could collect the the water there and absorb it and prevent it from running into bodies of water. Or they could be added sort of similar to mulch, only on the opposite. They could be added underneath fertilizer and absorb the water before it was left to run off. And then for future research, I would like to test the effects that pine pellets has on other chemicals in fertilizers, such as phosphates, because if it had such a significant change on nitrate levels, I'd be interested to see what it could do with other chemicals. Or I would like to change the experiment design to be more similar to the agriculture field, because I did have, my experiment was just in single bottles, and it didn't exactly mimic the agriculture field, and I'd want to see what happened to pine pellets and the other components when they were exposed to the elements. And then I would also be interested in testing the unprocessed pine on chemical levels, because since pine pellets are a processed pelleted form, I'd be interested to see what natural pine could do. And references.
Thank you. All right, Reagan. So how did your review of the methods used by scholars in the field inform your selection of your research method that is aligned with your research question?
After researching, like I mentioned with the grass buffer strips, it helped me to create the idea that pine pellets could be added similar to the grass buffer strips along areas where the water would be run off. And like I mentioned with the engineered bioretention, since they had showed that the newspaper had the most significant change, I wanted to add newspaper to my test. However, it did not show for in mine to be the most efficient.
Okay.
Reagan, how did the limitations of your method or data influence your new understanding?
The limitations were that I could not implement my components out into the agriculture field, and so I think that there could be limitations there because it might not essentially act the same if it's exposed to the elements, and it would not be fully proper out in the field.
Think back to the initial curiosity that sparked your inquiry. What other curiosities do you have now, and how has this process prepared you to explore them?
I was interested in originally red tide and just agriculture, and so I think that in future I would like to continue further with the pine pellets idea and testing them, the effect that they had on other chemicals in runoff, but I think also just keeping on the same aspect of red tide or eutrophication, just different imbalanced chemicals adding to water would help me with my research.
Good. Thank you.
Thank you.